at table 13.4, right before the role of viruses in cancer.
Well, it's up kind of there, so I don't really know. For latency of animal viruses, the replication of animal viruses.
Here's where you thought the two level you got.
Oh, maybe I didn't. I took out a page or something.
I think you probably skipped it, so you can show us that
Oh.
one
Yeah,
that you
this-
added. Yeah, I think you probably skipped it. so
Yeah, I
you can
added
show
this
that.
one in there, so you stopped right here, right?
Yeah, I don't have anything highlighted for that
Oh, you're right.
Okay. All right, that's what I thought. Choose everything. Wow. Make sure you guys sign in because there's extra points for you. All right. Latencies in animal viruses is similar to like sogeny and what? To what? Like half the class over there. Like that. It's similar to lysogeny how? The vocal half is not used.
Well, I don't know what misogyny is.
Lysogeny is that same replicative cycle where the virus gets inside of a cell and sort of hides for a while, and then lets the cell replicate. Remember that?
Maybe it hides in the cell and doesn't do anything. Latency, because latency means like.
Remember this?
Oh, okay, yes.
Virus bacteriophages can integrate into it. Latency is similar in humans. Like I said, I've had chicken pox. That means I have what inside of me?
Oh, shingles.
You know, I have the latent virus that's inside of me, right?
Mm-hmm.
Hanging out inside of me. Something's going to trigger it. Usually it's going to be something like stress. Something that's going to make you a little immunocompromised, and it's going to come out. The latency is basically the same thing. When animal viruses remain dormant in the host cells, you need to know provirus, guys. Because it's a prophage when it's just the bacteria just the bacteriophage in a bacteria that's going through lysogeny. In this case, it's called a prophage, provirus, or a latent virus. And you guys will see this when you get to clinics and things, because this is the new way they look for what? Back in the past, if you were going to test for HIV, how long did you have to wait?
What was that?
But how long ago? So let's say I thought I had an exposure right now.
Years.
Like 10 years, you told me.
No. You want to die? Hold on. Before you get tested. Not before it came on. It's about 10 years, yeah. Or if you don't get treatment. But how long did you have to wait back then before you could get tested?
Was it like 2 weeks?
Hmm?
What about two?
Two what?
Years?
No, you get tested before that.
2 weeks.
fortunately. But it was a couple months back
Oh.
in the past. At least 6 weeks, maybe even longer than that. That's a long time to wait before you get tested because they used to look for your production of antibodies. That's what they used to look for. Then they came up with a test that looked for a protein that's made by the virus.
Oh.
And that was another ELISA they looked at. That was about 2 weeks.
Oh, okay.
Still, that's a long time, right? I had a friend who had possible exposure. He was a surgeon. And he said that was the, see what do you do? Can the after test you get after 6 months? He said that was the longest 6 months in his life right before that.
Oh my God.
Now, they use a thing called PCR, which I want to do in lab at the end of this semester for you guys, because this is what you guys have been getting into. They're getting away from streaking plates and stuff like that. They're going to what's called molecular biology. PCR looks for the provirus itself. Because what is that provirus in HIV? But what is it though? All it really is what? Virus gets inside. Remember that? We talked about the virus gets inside of the cell. What does it do? And then it gets inside and then it uncoats, right?
Mm-hmm.
And then once it uncoats, what's left?
DNA.
Just the nuclear material, right? And the problem with retroviruses is they turn that to double-stranded DNA, and they find your DNA and they integrate it into your DNA. So really all the virus is once it's inside of you is what? DNA. So that's what you look for, and that's what PCR looks for. So now they guarantee they can detect it within about, they say 7 days.
Oh, wow.
There's some people that say they can find it almost that day. Maybe the next day after that. Because they're actually looking for the virus itself. And it's incredibly sensitive. Anybody here ever done anything with PCR? Do you have?
Where's the
Our
ball?
biology with that,
With Reyes?
yeah,
Okay, so you kind of know the principle behind it? Yeah. So we'll show you how we can use it for the, you guys are looking for mutations, right?
yeah.
I'll show you how we can use it to study stuff like this. Okay, maybe prolonged for years with no viral activity. I got the chickenpox when I was a kid. Thank goodness. I hope this is real wood. I haven't had shingles yet.
Thank God.
Okay? Some latent viruses do not become incorporated into the host chromosome. Write this down because it's a good test question that pops up for people. If you remember in lysogeny, the bacteriophage, the prophage, always incorporates into the DNA of the bacteria. Always. With latent viruses, some do, some don't. Retroviruses do, some of the other ones don't.
And misogyny?
They always do. The prophage always incorporates into the DNA of the bacteria. There's really nothing else inside of that thing, right? All right. If a provirus does incorporate, like HIV, it's permanent. You cannot get it out of that cell. So once HIV infects the T cell, the uncoached, reverse transcribes into double-stranded DNA, incorporates into that genome of that T cell. It is there for what?
Ever.
Ever, and that's why they cannot cure HIV right now. They can knock the viral load down with the medications down to what? Somebody ever? To what?
I think it's something to expand the lifespan, right, but
You can, yeah, they think some of them now, except there's some weird things coming later on. But sort of the idea, they think the lifespan now is almost the same as a non-infected person. However, there may be some medication issues that are coming up later on, but that's a whole different story. But yeah, great. I mean, from 10 years to 70, 75, whatever. For you?
Well, back to the medication. My understanding is that it's not killing the virus, but it like maintains it not to like replicate.
It stops it from replicating.
Mm-hmm.
It's okay. Let's go back, guys, because this is important with this. The new hepatitis C drug, right?
Mm-hmm.
You take that for, I think it's 2 weeks, maybe, some people? Maybe, some people, is it 2 weeks? We'll say 2 weeks. The virus is gone, right? Because the drug inhibits replication of the virus, and the virus can't replicate. So after a while, it tries to, it dies off. It's gone. You're cured, right? The medications for HIV prevent viral replication as well, but since it's incorporated into the genome of some cells, is it replicating at that point?
No, it doesn't need to anymore.
No. So the drugs can have any effect on that? No. So it's just hiding it, and that's the problem with those type of things. Yeah.
I know right now you're talking about shingles. Is there any reason why people would get it at a younger age, and what makes that virus come out? Or
Usually they think now the main idea is that if you get a little bit of enough immunocompromisation, your immune system gets a little compromised.
that could
That they think the immune system usually is able to sort of just, it'll kind of start to replicate and the T cells will kill that, so you just get back. You know, if you get a little immunocompromised and you can't keep it
become
suppressed,
active.
yeah, it pops up.
Okay.
Yeah.
So is
And
that
the immune cells pretty fast at taking it out. Yeah.
is that why with AIDS they do a cocktail of different kind of drugs? Like one is to stop the replication, and then I mean,
They
what
all
else?
stop replication in some phase of that.
Yeah.
The reason they do a cocktail is the odds of any virus, any particular virus, developing mutations that are going to make it resistant to 3 different drugs is pretty slim.
Okay,
Yeah. Because remember,
so it's just about resistance. It's not so much
it's about resistance.
eradication. Okay.
Right. The other ones all work pretty well.
Oh,
Plus,
okay.
It also, I mean, probably both. I mean, if you attack it in 3 different ways, you're for sure going to suppress it down. Because what they do now, that's what I'm talking about, they can get the virus down to undetectable levels in the bloodstream. That's how well the drugs can work.
Is that now why they're saying that it's okay? Well, not completely okay, but if a woman has AIDS, she can have a baby without the virus.
Yeah, they're pretty, they still like to maybe put their child on medication
Yeah.
first, but yeah. And that's what they do. In fact, I would put on that new drug actually. But anyway. So anyway, we're like,
Sorry.
so it's permanent now, and that's the problem. They're trying to find drugs that'll make it come out of latency. Because if you drive all of them to come out of latency, the cocktails of drugs should eventually get rid of them, right? They have not been successful in that yet. So that's the problem with it right now. And you want a good example of that? Who's our good buddy who just went down to Mexico to get treated for HIV? That actor, the irritating one. They're all irritating.
Oh, the tiger blood.
Charlie Sheen.
Yeah, tiger blood.
Oh,
Yeah.
And so he's HIV positive, right? You know that.
Of course.
So he, surprise. So he went down. He was on the immunosuppressive drugs. Not immunosuppressive, the antiviral drugs. His viral load was undetectable. What they tried to do in the past is before, in the late 90s and even the early 2000s, you had to take a handful of pills. And I knew a guy who said he didn't even want to eat because he was full on pills all the time. Now, how many pills do you have to take? Just one. They've got all 3 cocktails into one pill. So it's not that bad. So back then, they thought about letting these patients get on what they call drug holidays.
Oh, yeah.
The side effects were pretty rough.
Mm-hmm.
So they said after a year or so, they'd let you come off the drugs for a few months. And they were also hoping that maybe, just maybe, while you were suppressing the virus and your immune system was rebounding, maybe you were going to develop antibodies that would treat it.
Mm-hmm.
It didn't happen either way. In fact, it almost comes back worse. They said it would come back.
Wow.
So that's the problem. And that's what I know Charlie Sheen. He went down there to get on that medication, and like a week, 2 weeks later, bam, his viral load shot back up.
Wow.
Yeah, it couldn't happen to my guy. All right. Virion assembling infection outcomes. So virulent infection.
Did you add that one?
No, I'll put this in a little later, so you might have to write this part down.
Okay.
Or I can add it later if you guys want. But I pulled it from another book, so I like this explanation here. Virulent infection is lysis of host cells. This is the most common. If you get a cold, what are the symptoms of your cold?
Aching.
Inflammation.
Fever.
Just a cold, just a regular cold, not
Oh.
the bad, not the flu, just a cold.
Sore
What are
throat.
your symptoms?
Runny nose and a cough.
Runny nose, just irritating, it's all red. That's just the virus is replicating, lysing the cells, and your immune system is going after it. A latent infection is the viral DNA exists in the host genome, and the virions are not produced. What that means is it's just staying latent. It's hiding inside of there. Okay? Host cell is unharmed unless or until virulence is triggered. With shingles, it's hiding in your nerve cells, right? Is it doing any damage right now? Is that chicken pox doing anything to me right now?
No.
When I get, oh my God. If I get shingles,
Don't jinx
Oh, yeah.
yourself.
if I get shingles, is it going to start damaging my cells?
Yes.
Yes, it goes right back into the lytic stage, the virulent stage. It starts lysing those cells again.
So another one that would do that would be like herpes and.
Herpes, actually chickenpox is in the herpes.
Oh, that's
family. So yeah, a lot of those herpes viridans, they like to go to latency. Persistent infections, just for people, that's why I like this, are different than latent infections. Okay? A persistent infection is release of virions from the host cell by budding, does not result in cell lysis. Okay? So the cells aren't being damaged, they're not dying. Infective cells remain alive and continue to produce virus. Okay? These things turn into little what?
Factors.
Little factories. They just sit there and crank out virus. Okay, so that's a persistent infection. Different than latent. Transformation, on the other hand, is what? What does that mean?
Overgrowth in one cell.
So what can do it? What viruses can do this?
There's a STD that does that, right?
Yep. What viruses can do this, guys? We'll talk about it in a little bit. We'll get to it in a little bit. Just remember that certain of the viruses can transform cells into cancer. There's 2 different mechanisms. They can do it directly.
Oh, HPV.
HPV is one, and
Yeah.
indirectly. In fact, all you people out there, FDA just changed the rules on Gardasil. the recommendations for vaccination. So they bumped it up to 45.
What's Gardasil?
The HPV vaccine.
Oh,
The original recommendations were originally it was for girls under 13, right?
yeah. And boys too, no? Because
Then they
my
put
son.
boys in it later. It was just girls first, yeah.
Yeah, they made my son get it.
Yeah. And we have pretty scary 13 that's just I don't have daughters.
Oh no, it's
I
13
just got her to sleep.
Huh?
I just got my daughters at like
Ugh.
6 months ago.
And good luck. I mean, if I had daughters, I would have hated you. I've been sitting there with the shotgun and the boyfriend. It would have been terrible. Anyway, then they moved it up, I think, to 18 or 20 or something like that. The idea is they want to try and get you vaccinated before you've been exposed.
Mm-hmm.
Well, no, I think it's like anyone below the age of 27.
20 to 27, they just moved it to 45 now.
Okay.
What?
Why?
Because I think the idea is maybe the vaccine will help
Keep it
treat
out.
if you get a persistent infection. Maybe the vaccine will help go after that eventually, or maybe when we're exposed.
Mm-hmm.
Everybody, I'm getting myself in trouble there.
I don't know.
Most people who, unless you are very, how am I gonna say this?
Having fun?
Unless you have, I'm gonna get myself in trouble. No, I'm not doing anything. Unless you've been a very good nun, you've been exposed to what? HPV, more than likely.
Wow.
Odds are.
Even if you've only been with one person your whole
Well,
life?
had that one person only been with one person.
Oh, no. Hell no.
That's the problem. That's
Yeah.
the only circumstance you can have is if you had two people that you were completely monogamous and you all were, that's about the only way.
Yeah.
But the difference is there's 100 strains of HPV. There's only about 4 that actually can lead to cancer. So I think what they're thinking now is you might as well vaccinate these people because maybe they haven't been exposed to that one of those yet.
And in my experience, well, when I was a teacher, the people that I knew that got it, they would open up to me, were single mothers. For some reason, it was like weird.
Yeah,
And they were
vaccine?
always telling me, no, they would get HPV,
Oh, they get HPV?
and have to go through treatments, and it's like,
They're lying.
I don't know, but they would be like all scared about having cancer, and I'd be like, oh,
You just have to keep an eye on it.
that's kind of
But the
weird.
thing is, but that's the problem with HPV. How many people were symptomatic for it?
That's what.
A lot of people are asymptomatic for HPV. They get the virus. Most people get the virus and clear it. They don't even know they have it.
Yeah.
Yeah,
They've been
but like
exposed.
a year
Yeah.
and a half.
Some people end up getting symptomology, getting the warts and things like that from it.
Oh, that's how they know.
Some people get
Oh.
that, and then they clear that virus. And then some people get this persistent infection with it that they can get. It sticks around.
Wow, okay.
All right. Comparison, guys, just look over that. The differences. Just know the difference. What's a bacterial virus called?
Bacterial virus, phage.
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 That's part you need to know. So cell division is, we're talking about cancer. Cell division is under strict genetic control. Genes dictate some cells can no longer divide at all. Your nerves, right? Are not supposed to divide at all anymore. I know that's not 100% true. But we're going to say this. They do find that some of your brain neurons actually do replicate a little bit.
Oh, wow.
Cells that can divide are prevented from unlimited division. Do you all remember telomeres from 1306? What are they? Good, I remember the name.
I know they're like some sort of part of the
They're on in your chromosomes. They're little things on the end of your chromosomes because every time your cells you copy your DNA, you chop a little piece off. So telomeres are at the end because you don't want to start chopping off what? Stuff you need, right? And the idea is when you're younger, you have an enzyme that replaces those telomeres. It's called telomerase. After you stop growing, telomerase gets shut off, and every time your cells replicate, a little piece gets cut off. And eventually you start getting to what? So this is your chromosome right here. And these are your telomeres. Replication, 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 replication.
Oh,
Uh-oh, now what?
no.
I started getting rid of stuff that I need, right?
Right.
That's part of the aging process.
Oh.
And when you start, that's one of the things that cell's age. And it's sort of a It's sort of a safety factor. Because uncontrolled cell division is cancer, right?
Oh, yeah.
So if cells started uncontrollably replicating and they went out of control and became cancer, ideally, eventually what would happen?
You die.
They would die because they would lose all their telomeres, right? So it's kind of a precaution against that. I'll tell you why it doesn't work so well in a minute. All right, genes for cell division are turned off. These are called pro-oncogenes or oncogenes, right? And genes for inhibiting cell division are turned on. Those are called tumor suppressor genes. You guys remember any of those from 1306? Y'all remember P53? Okay, P53 is mutated in about 50% of all cancers. And P53 is sort of the master regulator. After DNA is synthesized, the protein P53 goes and scans your DNA to make sure there are no what? There are no mess-ups. And if that little last second guard gets mutated and that protein doesn't work, those mutations can get through. So about 50% of cancers have a mutated P53. Those are called tumor suppressor genes. Okay. Neoplasia is what? I just feel like I want to. I just finished the chapter on cancer. Neoplasia is cancer, malignant cancer. What does neo mean? Neo, what does plasia mean? What's hyperplasia? Growth. So neoplasia means new growth. Okay? Uncontrolled cell division in multicellular animals, and a mass of neoplasia cells is a tumor. And so you think about this telomerase, we may get tumors. So there's two things that may happen with us if you get a tumor, right? A, your immune system may detect it and get rid of it, your NK cells and everything else. Or B, it may start dividing, and after I think there's 20 some odd number of cell divisions before you start losing your telomeres. And it may start dividing and also loses telomeres, right? And then that cancer cells are going to what? They're going to die too. All right. What's the difference between a benign and a malignant tumor?
I think it's like the rate of growth.
Is that part of it?
Cancerous, of course. Like malignant is cancerous.
Malignant or what? So what's the difference? Benign tumors. Okay, we're going to go to a little bit of cancer. We're already finished with this chapter. Malignant and quote benign. Benign tumors are an overgrowth of cell in a particular tissue. But they still do not undifferentiate. They still look like that tissue. And they still kind of have function. Right? They also do not invade other tissues. They stay in that original tissue. And they usually become encapsulated. So it's like this little sheet and cover around it. So they grow for a while. Sometimes they stop. Sometimes they even start regressing. But the point is they don't metastasize. They don't invade other tissues. And they don't lose all their function. Does that mean they can't be dangerous?
No.
No. The only way they can be dangerous, though, is how?
If they grow in a bad spot, like your brain.
Right, that's the problem. There's one called a meningiomas. Anybody ever heard of that one?
Yes.
A meningioma is usually a benign growth in the meninges in the brain. Doesn't do anything unless it were to keep growing.
Takes up space.
And then what's going to happen?
You die.
There can be a little bit of root problem in the brain, right? So that can happen. Or you also hear about these kids. For some reason, they seem to come from other countries. I don't know why. I think they're the ones that make the news. Where doctors have taken out a benign tumor the size of a basketball out of an 8-year-old. I'm like, holy moly, how did that kid not know he had that in him? So are those going to damage them?
Yeah.
They can, because they can push against other organs. They can block up blood supply, things like that. But the tumor itself isn't going to do anything. Malignant tumors, on the other hand, undifferentiate, which means they're no longer functional. They don't even look like the original cell anymore. They start growing out of control and they start invading neighboring tissues. This isn't even metastasis. They actually start putting these little crab-like, they call it, extensions into other tissues. They don't function. All they want to do is what? Grow. They also, a lot of times, can't go through cellular respiration. They stop at glycolysis. But if they're growing fast and they're only making 2 ATP per molecule of glucose, that means they got to be burning a whole bunch of what? Okay, let's say your car, you've got a nice car that's running on 6 cylinders, and it gets 25 miles per gallon, right? For some reason it breaks and you got one cylinder, and you're getting 2 miles per gallon. You're gonna be burning a whole bunch more what?
Gas.
And that's the same thing with tumor cells. They are just gobbling up oxygen and glucose. So they also do what's called angiogenesis a lot of times. They start making new blood vessels so they can start stealing all the blood supply nutrients from surrounding tissues. And they can actually start starving the tissues that are around, because they're stealing all that, because all they want to do is grow. Okay? And then the last part about it is they can metastasize. And that means little pieces will break off and travel through the lymphatic system or the circulatory system and lodge into other tissues. In fact, most people who die of cancer die from what? Did they die from the primary tumor?
No.
No, they die from the metastatic tumor. Okay. Now, how come these things don't die from loss of telomeres?
Because they're getting fed.
They're getting fed, but they're dividing. Bam, 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 bam. In fact, you can't even detect a tumor until it's gone through 30 cell divisions, which means it's up to about 1 billion cells before they're even detectable. So how come they're not dying?
Is it because they're making new ones?
They actually turn telomerase back on.
Wow.
So every time they replicate, they rebuild their telomeres. So they become what we call immortalized.
So with kids, is that why like a lot of kids like across seas too? You see, they're always the ones that have these big tumors, is because they have that the enzyme that keeps continuing the ends of the chromosomes.
Telomerase?
Yeah. So that's why kids usually have like the bigger tumors, right?
Mm. That might be part of it. They turn that on.
Wow.
Because if a
That's
cancer doesn't turn on, eventually it should die eventually, right? It should reach the end.
But if the kid keeps continuing like you're saying, then they're never really gonna die.
Right, but they don't die. But there's that lady there's a lady back in the 62, 64, don't quote me on that, named Henrietta Lack. Anybody ever hear about her? She went in for ovarian cancer. The doctor kept some of her cancer cells. They've been doing research on them ever since. They're called HeLa cells. So
What?
I used to work on them down in UTEP. So how many cell divisions have these things gone through?
Can we reactivate
Trillions.
older people?
Well, okay, that's a good question here. So let's hold on to that, because I want to bring that up.
Okay.
So, now the HeLa cells aren't very good cell models anymore because they're rapid mutations. But they're what they're called immortalized cells. And we'll talk in a minute about how you grow these viruses. So they become immortalized because they turn telomerase back on. All right. So, how does cancer happen? They have what they call the multiple hit theory now. We won't go into that too much. But what's going to happen here? This is how a virus is going to do it. Okay? Here's a normal DNA. This is a proto-oncogene. Proto-oncogenes are genes that were originally designed to let cells divide and proliferate. And then once you get older, they get shut off, right? But they're still what?
There.
They're still there. Okay. So you're going to make a repressor, a tumor suppressor gene is going to make a repressor molecule that goes back and shuts these things down. We don't want to turn these on, we don't need them, so you shut them down. Okay? So the first hit, DNA, what happens? A virus comes in, remember we said viruses insert things in your DNA? Do they insert them with it? Do they have a special place? Do you have a little place in your DNA? This is where viruses go? No, they go to little places and they just stick them in there.
Wherever they.
Wherever they want.
Okay.
Or it happens a lot of times. In this first hit, what happened? A virus comes in and inserts a viral promoter. And promoters are what? They tell the RNA polymerase this is the beginning of the gene, right? This is where you start transcribing. Now, do viruses replicate very quickly or very slowly?
Quickly.
Very quickly. So these viral promoters are what we call high copy number promoters. They're constantly letting RNA polymerase attach and transcribe the gene. But you still have a repressor molecule here. So do you get cancer? No, you don't worry about it. You're fine. That RNA is attaching, but it can't do anything. RNA polymerase attaching, it can't do anything. The second hit, the virus inserts into the repressor gene. So now you've got viral DNA in the middle of this gene. So when this gene is transcribed, does that protein function anymore? Does that repressor molecule work anymore? So it's made, it gets degraded properly, but what happens over here now?
It's allowing it to continue growing.
So now you got rid of the repressor, so these are proliferation genes, and you've got a viral promoter in front of it. So they're just doing what? They're just turning that gene on, and it's just transcribing and transcribing and transcribing and transcribing. So it's just, it's like a perfect storm, right? Okay. Yeah.
What if it doesn't get hit the first time? Or does it have to get hit the first time in order to get into the repressor?
Not necessarily. You could, the first hit could be actually inside of this repressor molecule too. But these proto-oncogenes, even though they're being repressed, they're still not turned on that much. It's the bad part is that viral, like I said, that viral promoter is having that viral RNA polymerase is attached to it. So it's like full blast, right? Everything gets turned on full blast. Actually, it's called a 3-hit model now, instead of the 2-hit. But this is just with viruses. We're talking about cancer in general. It's called the 3-hit model. All right. Viruses cause 20 to 25% of human cancers. Okay. Some carry copies of oncogenes as part of their genome. So now what are they going to do?
They can insert it anywhere.
So now they're going to insert that dang gene in there and turn it on automatically. That's a problem. Some promote oncogenes already present. That's what we were talking about there. Some interfere with tumor repressor genes. That's that p53.
Oh.
They go in there and they get, and so if you have a p53 in the tumor and the virus inserts its DNA right in the middle of that p53 gene, now you've just done what?
Oh.
You've ruined p53. That protein's no longer functional.
And this happens because it's so random wherever they put
Yeah.
themselves.
They're using sequences that go in, but a lot of those sequences are right there. But it's random, basically. Yeah.
Wow. Okay.
All right. So some specific viruses, we know about 15% of human cancers. They think about 20, 25%. We'll go through a couple more, right? Yeah.
Sorry, so
Nope.
some are already in the host, like right there, promote oncogenes already present in host.
The oncogenes are already present in your host.
Some-
So those genes that were turned on for growth when you were a kid,
Mhm.
they get turned off, right? Because you don't want to be growing anymore. Those are the oncogenes, proto-oncogenes. It's a really confusing terminology. When I first took it, I was like, I don't understand why we have these genes and they're bad. And that's what it is. That's what I call them that. So what's a Burkitt's lymphoma?
Well, lymphoma's in your blood, or has to do with your blood.
Lymphoma, isn't it like the fat in your like skin, isn't it?
No, that's lipomas. That's close. That's lipoma. I got that.
That's the
We know way too much about that.
lymph nodes.
Lipomas are a little fat. I'll talk about that one in a second. It's not in your lymph nodes. Okay, you have leukemias and lymphomas, right? They're both blood cancers. What's the difference?
The lymphoma's in your bone marrow. It comes from your bone marrow?
That's leukemia.
Oh, dang it.
Leukemias are when the stem cells in your bone marrow become cancers.
Yeah.
So they grow inside of there. Lymphomas are when those white cells get out and become cancerous. So your B and T cells become cancerous. And they end up in the lymph nodes because that's where they hang out, right? So if you have a cancerous B cell in the lymph node, it's going to start replicating, and that's why the lymph node gets so big. Burkitt's lymphoma is from Epstein-Barr virus. Who here knows Epstein-Barr virus? Anybody here ever had mono? Am I the only one? Nobody here that ever had mono?
No.
Dang. How many people here have been exposed to Epstein-Barr virus? All of you, all pretty much, yeah. Most people don't end up getting it. I'm potentially at a higher instances of Burkitt's lymphoma because I actually was symptomatic for it. But it's still a really small percentage. What's Hodgkin's disease? That's another lymphoma. You've got Hodgkin's lymphoma, and then all the other ones are called non-Hodgkin's lymphoma.
Yeah, I've heard that. Why?
Because this guy named Hodgkin's discovered the first one. That's what they call that Hodgkin's lymphoma. And now all the other lymphomas are non-Hodgkin's lymphoma.
Oh my God. Is it like a?
Yeah, exactly.
Is it special? Is it? I mean, from other? Or?
Actually, if you're going to get a lymphoma, this is probably the one you want. Problem with lymphomas is, I actually had a relative who passed away from lymphoma, is some of them are incredibly treatable, other ones are not treatable. And some of them are very fast growing and other ones are very slow growing. And if it's a slow growing lymphoma, how are you going to treat it? Because how do cancer drugs work right now? What do they kill? Rapidly dividing cells.
Yeah.
So if you have these slow growing lymphomas,
Oh, it won't even catch it.
the chemotherapy drugs don't work on them. So, but you're talking 20, 30, 40 years. They're kind of weird, but Hodgkin's is pretty treatable, actually. It's one of the ones that's pretty treatable. It's susceptible to it. What's carposiosarcoma?
Sarcoma's cancer of the skin.
Mm-hmm. Caused by a virus, and who used to get carposiosarcoma? HIV patients. It's one of the indicator diseases for AIDS at the beginning. Because most of the time, most people with a healthy immune system can get rid of the virus that causes it. And if you don't have a healthy immune system. So the other people who get it sometimes are transplant recipients. Can sometimes come down with it.
Immunocompromised.
Yeah, if you're really severely you gotta be severely immunocompromised. Cervical cancer. Is that the only thing, the only place HPV causes cancer?
Uterine cancer.
Why do they worry about it now so much? Why do they want to start giving it to boys now and girls?
Because boys are the carriers.
See, you just make us sound like really dirty.
I have one. He
Well,
is.
isn't just being a male, you already have a higher chance of getting cancer? You just did that.
Well, they
No,
don't develop the symptoms. They give it to girls, and they can give it to more girls than. I have a 15-year-old. So.
No, that's part of it, yes.
Okay.
Considered carriers, yes, that is part of it. The other part now, they found out that HPV does not just cause uterine cancer or cervical cancer. It can cause vaginal cancer, penile cancer,
Wow.
head and neck cancers, throat
Oh,
cancers.
oh.
So, any mucous membrane now, they found out they can come in contact with HPV has the potential to cause cancer.
H.
So now haven't you seen the ad where it's got the little girl in it and she's an adult and she has it and they back it down and the next one's the boy?
Yeah.
Did you know that?
Yeah.
Yeah. What are some other ones that can cause by viruses? These are direct acting viruses, most of these. They're going to go in, they're going to interfere with the DNA like we talked about, and they're going to do some damage. What about liver cancer from hepatitis B and hepatitis C?
Oh yeah.
Sometimes they think that those cancers are not directly caused by the virus, they're caused by the chronic inflammation from the immune system going after the virus in the liver. So 2 different ways you can cause those things, direct and indirectly. So, what do I have here? Sometimes I really want to use this slide 3 in the morning. All right, so formation, okay, the proviral state and transformation of a tumor cell. So here's a cell, gets infected with a what? A virus. Viral multiplication, you're going to get what? Virus present, but not what? Replicating, that's a latent infection. That's chickenpox, right?
Yeah.
Now come down here, slow release virus without causing death, that's a persistent infection, okay? That's going to be something like hepatitis C, right?
Those chronic stuff.
And then these may revert back to lytic at any time. Now you've got this one right here, you've got lytic stage, death of the cell, release of the virus, that's the lytic stage, right? That's when the cells die. Now up here you've got what? Tumor cell formation and cell transformation, and this is where you get to what? Transformed into cancer cell. Alright, so viruses cannot grow in standard microbial media. We cannot grow them on a plate, right?
Mm-hmm.
Even though I went to a science fair and I judged it. Oh, I love fraternities so much. We went to the science fair in Lincoln, what they call it back then? Junior high, I think they call it back then. And one of the local fraternities from Utah was the science fair judge, and I was just looking at it. Girl who won sweepstakes had a bunch of petri dishes out there, and her title of her thing was, "How effective are household cleaning agents at killing viruses?"
Wow.
And she got sweepstakes for that.
Wow.
You didn't say anything, you're just like, yeah.
It was already overdone with by somebody else. Okay. Cultured inside of host cells, that's why you have to grow them, and that's why it was so hard to get anything for hepatitis C, because we could not grow it in cells for a long time. Okay? 3 types of media for culturing viruses. Media consisting of mature organisms, how about that? Embryonated eggs. This is where they grow what? Huh? But what viruses do they grow?
E. coli. No, that's
That's
a
a bacteria.
bacteria.
Anybody ever had a flu vaccine?
Oh, that's
What do they ask you every time
right.
before you have a flu vaccine? Are you allergic to
Dairy.
chicken? They
Yeah.
grow the flu virus in chicken eggs. It's nice, it's sterile, nice and sterile in there. You just put the cells in there, they just replicate, and they just have big old baths of these poor little chickens. And cell cultures. So culturing viruses, mature organisms, culturing in bacteria, that's easy. Does anybody care about bacteria?
No.
No. You kill all the bacteria, the bunch of phages, nobody cares. That's what I think we're going to try and do next week. We're going to try and do a phage display thing. We'll try. Phage grown bacterial liquid cultures on agar plates. Lysis of bacteria produces these things called plaques, and what I want to try and do is called a plaque assay. We'll see if we can get it to work. So this allows an estimation of phage numbers by the plaque assay. So what you do is you get a petri dish, an agar, and you cover it. You put what's called a lawn of bacteria. You don't streak it. You want the whole thing covered with bacteria. And then you're going to add the sample with the phages in it. And anywhere you get these little holes, that should have been what? One single phage at first.
Wow.
And as they replicate, they start killing the bacteria. So that little thing is bigger and bigger and bigger. So we're going to try and do that. See if we can do it. If you guys do it, I'll give you credit in our next lab manual.
Yay.
Culturing viruses in mature organisms, culturing viruses in plants and animals. Numerous plants and animals have been used to culture viruses. They're trying to do it right now with tobacco plants, actually. They have nice big leaves, they grow real well. Laboratory animals are difficult and expensive to maintain. How much do you think it costs to keep one mouse a UTEP in their thing a day?
$3.
Not quite that much, but it's pretty close. That's one mouse. If you have 1,000 mice in your colony, how much is that a day? See how expensive this gets? Why people are trying to get away from animal models? They really are. People, I mean, A, it's expensive. B, nobody really wants to have to do anything. We're trying to move away from them. Problem is, there's some phases where you have to use it. You just can't get around. How about from ethical concerns? That's another one. They're really big and really strict on how you treat animals now. They've gotten a lot better. How about humans though?
What do you mean?
Well, we're going to test hepatitis C drug on somebody. Who here wants to volunteer to get hepatitis C?
I mean, if they're on their last leg, people will be
No,
volunteering.
you don't want to last leg because their immune system's down. Everything else, you
Because
know.
I've heard some prisoners like
Yeah.
put in like they're like,
Yeah.
why not? I've heard prisoners.
So what about convicts, prisoners? I like the term convicts for this particular discussion. Why not? Why can't you use them for that?
Human rights.
Oh, it's gonna be-
Well,
I think it's fine. I'm just
What
saying.
if they volunteer?
Yeah, I think.
But what if they
Everybody's
get
different.
out?
People
What
are
if they, like,
different.
get out and like.
Well, we got, that's a problem maybe. Hopefully they'll get over it. But
You can spread it,
maybe spread it. But what if they weren't? What if you found something, you know, you kind of trusted, they volunteered, maybe a little extra TV time, maybe a little extra carton of smokes, I don't know. Why couldn't you do that? Why don't they do it?
Is it because everybody's different? Everybody.
Part of it, one, it's a particular population. B, a lot of prisoners already have things like hepatitis C and a bunch of other things, so they're not a good subsample of the general population.
Oh, the
C,
general.
the main reason is no matter what, they're still considered being under duress. Because they're gonna get more TV time, or they're gonna get more cigarettes,
like
or they're gonna get better food. or
Oh.
something. So because of their, so it's a big, huge thing, it's called the Nuremberg Protocol and all this stuff.
Oh.
So no matter what, you can't even do it. And D, the data is just not needed. Because a lot of them are already gonna have hepatitis C possibly, or something else. So it's just good, but the ethical part is, they are under duress, you know, they are gonna get maybe extra TV time, right, or some more better food, or more yard time, or something like that, that's not, so ethically, you cannot do that. How about college students? Anybody here wanna volunteer? Pay a couple extra bucks?
We're under duress.
You're under duress? Actually, you kind of are actually too with that.
Yeah.
A lot of college students have trouble paying bills and things like that.
Yeah.
So you have to be very careful with these things. Okay, cultivated viruses in chicken eggs are inexpensive. They're among the largest cells. They're free of contaminating microbes. They have nourishing yolk, which I don't understand what that has to do with the virus, but that's okay. Fertilized chicken eggs are often used. Embryonic tissues provide the ideal site for growing viruses because there's really no immune system, right, yet, because they're embryos.
Mm-hmm.
And the cells have all the nutrients they need and everything else, so they grow pretty well. Some vaccines are prepared in chicken eggs. This is going to be the flu one. All right. Poor little chicken.
Oh.
They really just have to put that needle right in your back.
I know.
Put a little egg or something.
Oh.
Okay, culture virus and cell or tissue culture. It's not tissue culture and cell culture are often confused. Tissue culture is when you're actually growing tissue. Cell culture is when you're growing cells, right?
Oh.
You grow them in media or broth. In cell culture, sometimes inactive. Oh, there you go. Tissue culture. Two types before we leave: diploid cell culture and continuous cell culture. So this is going to be the media right here. Oh, origin. All right. Oh. So, the difference between that is, are your cells diploid or haploid? Or polyploid?
I think did.
Better be diploid, you guys are something.
Okay.
You guys out there, right? So, this is what they call primary. This is another name. Write down primary cells. Okay. But what do we say about telomeres in primary cells? If you're trying to grow them in a flask, they have telomeres still, right? And they don't have telomerase turned back on. So how many times can you let them replicate? There's a finite number of times they can replicate before they start to die. Okay? Continuous culture cells are cancer cells or immortalized cells. Most of the work in these cells are done in different types of cancer cell lines because you don't have to worry about them dying off. You can let them grow and grow and grow and grow. Eventually, you got to move back though into something else because cancer cells have different properties too. Like those HeLa cells they talk about now aren't that good because they're like these Frankenstein cells. No. It's interesting because her family, you didn't hear about it in the news? Her family went back and sued because the physician who took her cancer samples never told her. He didn't ask for permission or anything. Now, I'm pretty impressed with the family because they won the lawsuit and all they asked for was all they asked for is that she got recognition for it.
Oh, that's
For her
good.
contribution to time. That's pretty good.
That's great.
So our virus is alive. I'll let you guys answer that question. Care to? Virus. I'll talk about this in lab. All right, I'll pass the test. We'll go to the test in lab. And then I'm going to give you guys a 50 ml conical tube that I want you all to find some soil.